What is high-resolution MELT? High-resolution MELT is used for detection of mutation, polymorphism, and epigenetic differences in double-stranded DNA samples. In order to perform this test, double-stranded DNA samples are needed. Meanwhile, polymerase chain reaction was used to amplify the DNA region in which their mutation of interest lies. This region of interest that is amplified is known as amplicon. HRM analysis process simply uses precise warming of amplicon DNA from around 50 degrees Celsius up to 95 degrees Celsius. At some point during this process, the melting temperature of the amplicon is reached and the two strands of DNA separate or melt apart as shown in this picture.
HRM process is monitored by using a fluorescent dye aka intercalating dye. They bind specifically to double-stranded DNA and when they are bound, they fluoresce brightly. At the beginning of the HRM analysis, there is a high level of fluorescence in the sample. As the sample is heated up and the two strands of DNA melts apart, presence of double-stranded DNA decreases and thus fluorescence is reduced. The machine then simply plots this data as a graph known as a melt curve, showing the level of fluorescence versus the temperature.
Now I would like to discuss on the analysis of HRM curves. How about normal DNA versus the mutated DNA? The melting temperature of the amplicon is dependent on the sequence of the DNA bases. The two samples from two different people should give exactly the same shape melt curve. However, if one person has a mutation in the DNA region, then the temperature at which the DNA strands melt apart will be altered. Since the two melt curves appear different, it indicates the existence of the mutation in the DNA region. So, what is mutation? Mutation is a permanent alteration of the nucleotide sequences in an organism. There are three possibilities of the mutation. It can be neither allele contains a mutation, which is known as wild type, or one or other allele contains a mutation, which is known as heterozygote, or both alleles contain a mutation, which is known as homozygote. Heterozygote mutation is characterized by the changes in the melt curve shape. For the homozygote mutation, is characterized by a temperature shift on the resulting melt curve.
There are many advantages of high-resolution melting analysis. Firstly, it is cost-effective. It is more cost-effective than other genotyping technologies, makes it ideal for large-scale genotyping projects. It is also have simple and fast workflow. After performing PCR amplification of the target sequence, no additional instrumentation is required for high-resolution melting analysis. It is fast and powerful. It is able to accurately genotype huge number of samples in a short time with high level of accuracy. It also have low reagent consumption. It requires only PCR reaction volume for sample analysis. HPLC solvents or DGGE gels are not needed. There are many applications of high-resolution melting analysis, including SNP genotyping, DNA mapping, DNA fingerprinting, mutation scanning, species identification, zygosity testing, and DNA methylation analysis.